Okay, so welcome everybody. So let me do some introductions before the speaker talk, right? So as usual, I will do a general introduction first. Okay. So today's speaker is Professor Dawei. So welcome everyone. His talk will be the versatile roles of type 3 effectors in Rhizobium-Soybean symbiosis. So this is the 30th talk of a series targeting to promote agrobiotechnology and enhance understanding of the potential applications of agricultural products. We hope the series will inspire international scholars, researchers, farmers, and business in the agricultural field, as well as the interested public. First, let me introduce our speaker, Professor Dawei. He is currently a professor and doctoral supervisor at Northeast Agricultural University, a Xiangjiang scholar, and has been awarded various honors, including the Young Academic Backbone of Heilongjiang Province and Northeast Agricultural University, as well as the Youth May 4th Medal of Northeast Agricultural University in the academic year 2017 to 2018. He is also a youth editor of the Chinese Journal of Oil Crop Sciences and a guest editor of Frontiers in Plant Sciences. In the past 5 years, he has decided over 15 national and provincial level products, won 4 provincial and ministerial level awards, including 1 second prize and 1 third prize, published over 40 SCI research papers, and authored 3 books. He has also cultivated 3 soybean cultivars with a cumulative planting area of over 500,000 hectares and obtained 2 national invention patents. His research focuses on the molecular mechanisms and applications of soybean microbe interactions, and he has conducted detailed work on soybean symbiotic nitrogen fixation and soybean disease resistance. In this talk, Professor Xin will introduce the progress and the prospect of rhizobial type 3 underlying soybean-rhizobium interaction, including the function of the type 3 effectors, the soybean genes interacting with type 3 effectors, and the soybean breeding prospect in symbiosis modifications. So without further ado, let's welcome Professor Xin to the stage. So Professor Xin, please share your PPT.
Okay, thank you, Tom. Okay, can you see my PPT?
Yes, you can see it.
Okay, let's begin. Okay, first, thanks to Dr. Lam's invitation. Today, my topic is about the versatile roles of type 3 effectors in Rhizobium-Soybean symbiosis. I'm Dawei Xin. I come from Northeast Agricultural University. Today, my talk will divide into 3 parts. First is what is the type 3 effector. Second, I will introduce something our research advanced about the type 3 effector regulated symbiosis on soybeans. And third, we'll show some primary works. Recently, we do the interconnected networks analysis of type 3 effectors. First, I will introduce some background. We know that soybean is a special legumes and is important for China agriculture and for the food security. We know that legumes can construct a symbiosis with rhizobiums, but we know that soybeans is a special one because soybean seed has a high protein content. But we know that rice is not have so many proteins in seed. So I think maybe it's one reason why that soybean needed to construct a symbiosis relationship with the rhizobiums, because that the protein synthesis required a huge amount of nitrogen and salts. So the soybean required rhizobium to help fix more nitrogen from the air to support the protein synthesis. As we know that in the air, there are about 78% of nitrogen, but cannot be used directly by the plant, and this rhizobium can be helped. So it's my research is focusing on the construction relation between soybean and rhizobium. In the previous result, human have found that when soybean absorb the nutrition from the root, we know that when the nodule fix the nitrogen can be transferred into urea, and the urea can be transferred from root to the shoot. And this some part of urea can be directly transferred into the seed and can be used for the protein and yield synthesis. But when the chemical fertilizer nitrate cannot directly used by the seed, it needed to transfer from the root to the shoot and in the leaf by the photosynthesis process and transfer this nitrate to the amino acid, and then the amino acid can be used by the seed. So it's required additional energy to transfer this nitrate to the amino acid. So it's why if soybean have selection, maybe the nitrogen is more good for the protein synthesis. We know that for the legumes, different kind of legumes can be formed nodules with different rhizobiums. Recently, people have found that there are two types of nodules can be formed between when rhizobium infection the legumes in metacargo and the common beans, or some strains also chicken beans. This is kind of legumes can form the indeterminate nodules, something like this. And for soybeans and pea and some more common pigeon beans, they can form the determined nodules. For the determined nodules, the nodule size can increase not depending on the cell size increase depending on the cell size increase, not depending on the cell number. But in the indeterminate nodules, they have special development room. In this room, nodule the cell number will increase. So for the indeterminate nodules, nodule size increase depending on the nodule numbers, but not nodule size. And in previous analysis, we also found that one single rhizobium can maybe infect different kind of legumes, such as NGR234. This kind of rhizobiums can form the nodules in different kind of legume. But in some legumes, it can induce the indeterminate nodules, but in some it can induce the determinate nodules. So this means that the signal communication is complicated between rhizobium and the soybeans. And now human confirmed consistent that three parts of signals exist is important for the symbiotic relationship construct. First is that flavonoid or the isoflavonoid can be secreted by the legume from the root into the soil. Once the rhizobium recognizes the flavonoid, and this isoflavonoid can be entered into the rhizobium and bind to the node D. Node D is the transcription factors, and once node D is bound to isoflavonoid or flavonoid, this protein will be activated, and then downstream genes will be activated. Some genes related to symbiosis will be activated, including node effectors and type 3 effectors, and some more other catein oligosaccharides, something like this. And for the node factor recognized, it happened on the hair root of legumes, and some receptors have been published in Metacargo and Lotus Genomics. In the model, it's like this: when the node factor is recognized by the receptor, and the the factor signal pathway will be activated. It's a symbiosis signal pathways. It's very conserved in different kind of legumes. But in some legumes, such as metacargo, it different for the rhizobium effector metacargo. There's no type 3 effector system. But in soybeans, human found that type 3 effectors secreted where type 3 secretion system is important. And recently, some type 3 effectors also found have function to interfere with the effector triggered immunity and the pattern triggered immunity in host. And some effector also can have other transcription factors in the nucleus to regulate some gene expression. So today, I will introduce some effectors research in our lab involved in the soybean and the rhizobium interaction. At the beginning, the type 3 secretion system was identified in the human pathogens, such as Salmonella and Yersinia and E. coli, something like this. Human found that during the infection of this kind of pathogens, the pathogen can embedded to the membrane of human cells. Once the pathogen embedded to attach to the human cells, some proteins can be secreted into the human cells to interfere the human immunity system to promote the pathogen infection and to amplify in the host cells. This inject like as you will use a needle inject vaccine. So this structure human think is like this. When the like a needle injected into the host cell, and once the needle injected into the host cell, the pore complex will be formed. In sometimes the effector proteins here we named as type 3 effectors will be secreted from this structure into the host cell. In sometimes this secretion system is required energy. ATP can support this energy. By insect the effector into the host cells, pathogen also form the fish swarm, something like this. The fish by the fish swarm synthesized, then in sometimes the pathogen will continually to secrete the effector into the host and to play different functions to inhibit the host immunity. But for the human cell or other animal cells, the immunity system will be formed rhizome to recognize this effector to inhibit the infection of pathogens. But we know that furthermore, for the transmission electricity microscope emitted, human can have the detailed phenotype analysis for the type 3 infection secretion systems. From this picture, we can see that in the similar cells at the surface, we can see that some needle-like structure can be found. And by increasing the resolution, a needle-like structure can be found. So this is the first time humans see that once the exact structure of type 3 secretion systems. By the different Salmonella or Yersinia or E. coli, these needles used as a pili can be also increase the different length. It can affect the infection efficiency of pathogen. So type 3 secretion system, also called the insectosome, is one of the bacterial secretion systems used by bacteria to secrete their effector proteins into the host cell to promote variance or colonization. Recently, a lot of structural proteins about the type 3 secretion system was elucidated, and the function of this protein is also known. So by the comparison, a human found that in the rhizobium, they also have some proteins homologous with these structural proteins, but not completed. It also may be some difference exists in the rhizobium. And by the progress of rhizobium genome sequencing, the conserved type 3 secretion system coding genes was identified. such as Rhizobia-Soybean USGA110 and PNGR S234 and something, and also this MS Rhizobium lote. But we also, as I said before, that in some special rhizobiums, there's no this kind of structure proteins was found. So at the beginning, human hypothesis that the type of 3 secretion system in rhizobium is like this, maybe, but now it's still unknown the exactly structure. But we know that NOP A and NOP B may be the needle structure for, and NOP X is the pole proteins. And also by the comparison of genome sequencing and for the type 3 effectors in different rhizobiums and other bacteria and pathogens, human found that they have special rhizobium type 3 vectors such as NOP-L, NOP-P, NOP-C, and NOP-E. Here we need to show that in rhizobium, the type 3 effector is called named as nodulation of the proteins. So all the effector in general, we named it as NOPs, so it's named as NOP-M and NOP-T. Then this kind of effector NOP-T, NOP-M, and NOP-D, NOP-F, these proteins, type 3 effectors, all have the homolog proteins in other bacteria, especially some pathogen effectors. So today, I will introduce several effectors of this kind of type 3 effector, NOP-M, NOP-T, and NOP-C, and NOP-A. First, let me introduce our research progress in NOP-M. By the overexpression of M in vitro, we purified these proteins. And by the function analysis, we found that when NOP-M have processes E3 ubiquitin ligase activities. From this picture, Western result picture, we can see that when combined with E1 and E2 and ubiquitin and NOP-M together, the polyubiquitinated linked proteins can be identified. So this means that NOP-M possesses E3 ubiquitin ligase. And by the animal acid analysis, we found the conserved region at the 338. This position animal acid is the activity animal acid for the NOP-M. So when we get the function of NOP-M, it has E3 ubiquitin ligase. We hypothesize that because NOP-M belongs to the rhizobium, is a prokaryotic cell. It has no ubiquitin systems. So we think that maybe these proteins can be secreted into the host cell to ubiquitate some target proteins, maybe involve the target protein degradation or other functions. So we think we first overexpress NOP-M in tobacco to see which signal pathway it was involved. By the overexpression, we first analyze the RS signal pathway at first. From the result, we can see that when NOP-M have the E3 ubiquitin ligase activity, the RS reaction was inhibited. But when NOP-M lost its function, the RS activated again. And also two relative genes also analysis by the qRT-PCR. From the result, we can see that in the tobacco, RS and RS related genes also have response to NOP-M. This supply a clue that NOP-M may be involved in the kinase or something immunity signaling pathways in host. But we know that tobacco cannot form nodules with rhizobium, so we must to elucidate a function in legume. So we transfer to the soybean. We select RNA-seq as a method to identify the candidate genes at the early stage, because in general, immunity is happened at the early stage. By the RNA-seq, we identify about 2,000 genes. In these genes, we selected the huge significant expression genes response NOP-M as the candidate genes for future work. So maybe this this genes will checked, but only the GMFP1 ones can be confirmed having directly interaction with norm by the pull-down analysis. In the same times, we also do the BIFC analysis in the tobacco cells. From the result, this result support us that the interaction between NOPM and the GMIP exists. Furthermore, we also do the luciferase analysis in tobacco. From the result, we can see that they have a species interaction between NOPM and the GMIP in the tobacco. So this result support us that GMIP can be directly interacted with NOPM in soybean. As in soybean, we want to know if this NOPM functions on the nodulation. So we check the exactly function of NOPM on GMIP. As the GMIP have the function as a E3 ligase, so we do the ubiquitinating action again to check if GMIP can be ubiquitinated by the NOPM. From the result, we can see that then GMIP together with NOPM and ubiquitin and E1 activity and the E2 activity kinase, we can see that the polyubiquitinated lens protein can be found at the GMIP. So this means that NOPM can ubiquitinate GMIP. This means that GMIP may be degraded by NOPM ubiquitinated and maybe I'll have new function by ubiquitinated. So we co-express these two proteins in tobaccos, and MG132 is the inhibitor of the 26S proteins. So we can see that when the MG132 exists, the GMIP showed a stability in the tobacco, but when without MG32, the GMIP showed completely degraded. So this means that maybe in the host cell or soybeans, NOPM can ubiquitinate the GMIP and degrade these proteins and to interfere with the host cell. So furthermore, we do the transgenic soybean plant to check if these genes have a reaction to NOPP induced nodule phenotype. At the beginning, we mutant NOPM in the rhizobium. From this result, we can know that when NOPM is mutant, the nodule number and the nodule drive rate will be decreased significantly. But when the GMIP is overexpressed in the soybean, you can see that all the nodule was inhibited. There nearly no nodule number can be found when inoculated with wild type Rhizobium HH103 or NOPM mutant. But when GMIP was knocked out in soybeans, we can see that more number and more nodule drive rate can be found. This supply a clue that maybe as GMIP is immunity related genes, so we think that when the symbiosis during the symbiosis establishment, the NOPM can regulate the degradation of GMIP and to control the nodule level, to control the immunity level at the normal level can be allowed rhizobium construct symbiosis. So the conclusion is that the effect of NOPM on nodulation is GMIP dependent, and too much GMIP is not so good for nodule formation. And by the RNA-seq analysis, we also found that PR5 is the R genes trigger immunity involved in the NOPM and GMIP involved signal pathways. So this signal we showed at here is like that. When rhizobium infects soybeans, the NOPM can secreted into the soybean cells. When the GMIP will be ubiquitinated by NOPM. When after the GMP degraded, the PR5 signal pathway cannot be activity. So these immunity levels will be kept at acceptable levels for rhizobium infection. Second, I will introduce another type of effector. It's NOP-T. By the in vitro analysis and protein purification analysis, we found that NOP-T have autoproteolytic activity. And by the homolog analysis, we found that NOP-T have homolog proteins in pseudomonas. It's named as ARIA-PPHB. ARIA-PPHB can be as a scissor to cut the BIK1 and to inhibit the pattern-triggered immunity. In same times, another protein PPS1 also involved this signal pathway to inhibit to active the effector-triggered immunity. So we wish to identify the proteins come directly with NOP-T in soybeans. So we use the IP combined MS analysis method to identify the candidate proteins directly interacted with NOP-T. So this is all the candidate proteins we identified from the MS result. Luckily, we found one genes also called the homolog protein with GMPPS1. So we selected these proteins as the candidate proteins for future analysis. By the BFC analysis, we found that NOP-T can directly interact with PPS1, and the transfer analysis also showed that NOP-T and PPS1 can interact with in the tobacco cells. Furthermore, we also do the pull-down analysis. We can see that NOP-T can also directly in vitro with GMPPS1. So this result support us that the two proteins can be directly interact, and the NOP-T maybe have a directly interaction with GMPPS1 during the rhizobium infection. Furthermore, we also checked the effect of GMPPS1 on the nodulation. So we use the hair root transformed system to check what's the function of GMPPS1 on the nodulation. So from the result, we can see that when at the wild type soybean cultivar 314, you can see that when inoculated with wild type rhizobium HH103, the nodule number is about 10, but when NOP-T is mutant, the nodule number increase to about 15. So this means that NOP-T maybe have a negative effect on the nodule formation. But when GMPPS1 is overexpressed, we can see that the effect of deficient NOP-T is disappeared. The nodule phenotype is same as the wild type induced, but when we inoculate with the wild type nodules, the more number was found. So this meaning that maybe GMPPS1 is a positive regulator for nodule formation, and the NOP-T interact with GMPPS1 maybe to control the rhizobium not to form too much nodules with soybeans. So we also do the CRISPR mutant. This result also support us that GMPPS1 have a response to NOP-T on the nodulation formation. Furthermore, we also do the GMPPS1 mutant RNA-seq to further mining the proteins may be involved in GMPPS1 signal pathways. So some candidate genes among the identified may be have co-expression pattern with GMPPS1, such as RPS2, RPS5, and RN4, and PPS2, and RPM1. And first, we select GMRN as the candidate proteins may be interact with GMPPS1. As in soybeans, genome redundant, we analysis the 4 homolog genes in soybean at first. By inoculated with rhizobium, we found that only GMRN4B have a response to rhizobium and can be expressed in the soybean root. So we only select these proteins as a future analysis. By the BFC analysis, we can see that GMRN4B and GMPBS1 have directly interactions. And furthermore, the pull-down analysis also support that these two proteins have interactions. And furthermore, we also do the overexpression hair root and the CRISPR soybean to check if these genes involved in the novelty and trigger the signaling pathway. From this result, we can see that GMRN4B also involved in the novelty triggered signal pathway and also have the effect on the novelty induced nodule number. So the conclusion for this is that GMRN4B is another protein respond to novelty, and in the same time we involved another type of effector is novelty. We can know that novelty maybe also have interfere with the novelty involved signaling. So here we showed a model. It's like this. During the rhizobium effect into the soybeans, NOP-M play as the ubiquitin ligase and NOP-T directly into the PBS1. PBS1 come directly into the RN4, and maybe NOP-P can also involved in this signal pathway depending on other proteins. Third is another type of effector is NOP-C. First, we construct this effectors mutant in rhizobium. By the nodulation analysis, we can see that maybe at the beginning infection, the crack or thread infection thread formation have difference due to the NOP-C mutant. So we performed the nodulation phenotype in about 310 soybean germplasm, including cultivar, landrace, and wild-type soybeans. There are about 200 cultivars and about 70 landrace and 40 wild-types soybeans. From this result, we can see that when NOP-C mutant, the difference was identified between cultivar soybeans and wild soybeans and landrace soybeans with wild soybeans. So we think maybe in wild soybeans, they have different genes, maybe respond to NOP-C. So we select Swinton-14 and another two wild-type soybean to further do the nodulation analysis. From this result, we can see that when NOP-C is mutated, some special different nodule phenotype was identified, and not as like Swinton-14 as inoculated with the wild type rhizobiums. The nodule number is 40, but when NOP-C is a mutant, degrade. But in wild soybeans, the difference is not so significant. So maybe during the dominant of soybeans, some genes diversity is happened for this for genes to respond to NOP-C. Because our lab have the genetic population is constructed by the Swinton-14 and the ZYD 0006. So we use the population to identify the genes maybe respond to NOP-C. And also we also overexpress NOP-C in the tobacco. We can see that NOP-C cannot induce the cell death. By the location analysis, we found that at the chromosomes, different chromosome segment substitute lengths have overlap fragment, maybe respond to the nodule phenotype difference. And in this region, GMCRP1 is genes maybe we think can respond to NOP-C. In addition, it's also a lot of other genes. First, we selected these genes due to the qRT-PCR because in this region not only one gene exists, so we use the qRT-PCR to detect if this gene's expression is induced by NOP-C. But finally, we only find that this genes, the GMA19G 176 T3 and 00 can respond to NOP-C. So we construct the genes RNI and overexpression high root to check the nodule number have difference when inoculated with wild type rhizobium or NOP-C mutant. From this result, we can see that these genes we named as GMCRP can have a significant response to NOP-C induced nodule. Furthermore, we analyze the genes haplotypes in our soybean germplasms, and 4 haplotype types was identified. As Swinton-14 and ZYD 006 is the parents of our genetic populations, and haplotype 1 and haplotype 2 was identified in these 2 parents. So we can think that, and also we found that these 2 haplotype also have a key function to affect the nodule numbers on soybean. So maybe in future, as we have the genetic populations, these 2 haplotype can be used for soybean breeding to regulate the nodule number and for some special cultivar. In the same time, we also use the IPMS method to identify the protein can directly interact with the NOP-C. Now here we identified this 8 candidate proteins, but only 2 can have directly interacted with NOP-C at the BIFC level. We can see that GM9 is a plant disease resistant response proteins, and the GMABHD is alpha beta hydrolase folder containing proteins. These 2 proteins can directly interact with NOP-C. So we also failed to construct the protein expression vectors to purify the protein to do the pull down analysis in vitro. By the result, we can show that GM9 and ABHD can directly interact with NOP-C. And by the overall expression of GM9 and the knockdown of GM9, the plant result so that these genes also have response to NOP-C. So here we give another model is like that. When NOP-C secreted into the soybean cells, this protein can directly interact with 2 proteins. One is GM9 and another is GMABHD. And by the RNA-seq analysis, we also found the PR5 also involved in the NOP-C trigonal pathway. So maybe these 2 proteins can have interfere with this signal and to affect the ETI or RS, something like this, to regulate the nodulation formation. Third is another factor named as NOP-AA. By the function analysis, we can find that NOP-AA can digest S-glucan and beta-glucan. So it's meaning that this protein have hydrolase activities. And by overall expression on the tobacco, and we also found that NOP-AA cannot trigger cell death. And by the nodule phenotype analysis, we found that when NOP-AA is mutant, some infection threat is different, and the infection event is decreased by the NOP-AA is mutant. So this meaning that maybe at the beginning NOP-AA, the hydrolase activity of NOP-AA can have an effect on the infector thread formation. So we also found want to identify the genes maybe have a response or can be directly interacted with NOP-AA. So we use the high generation real population to identify the QTL's response to NOP-AA. And this is the QTL's we identified in the soybean chromosome. And we selected several candidate genes to do the response, but only one gene at the 19 chromosome can be found on the one parents of Chalcedon cultivars. This gene we named it as the ARP. GM-ARP is not AA response proteins. By using the higher root systems, we use the ORI expression and kick out to see if these genes have response to NOP-AA induced nodule phenotype. From the result, we can see that when GM-ARP was overexpressed, the nodule number was increased, but it was knocked down. Nodule number is decreased. So it's meaning that maybe this GM-ARP also involved in NOP-AA, involved signaling pathway during the symbiosis establishment. Furthermore, we also analysis the haplotypes in the different soybean germplasms, and about 8 haplotype was identified in soybean. So on haplotype 1 and haplotype 2 was identified in the parents of high generation recombinant inbred lines. So maybe these 2 haplotypes can be used to modulate the soybean nodule numbers in future. Maybe this haplotype also involved in the infection activity at the beginning of rhizobium infected soybean. And from the QTL's identified result, we also find other genes is ERF5. ERF5 can directly interact with NOP-AA. And as by the homolog analysis, we found that GM-ERF5 is a transcription factors. So we think maybe when NOP-AA interact with these proteins, it can be affect the regulator of downstream genes. By the promoter activity sequence analysis, we found that GM-ERF may be regulated the promoter of GMPS1. So we cloned the GMPS1's promoter regions is upstream 3 base pair, 3,000 base pairs upstream of GMPS1's stack codon. So to perform the EMSA analysis, from this result, we can see that the GM-ARF5 proteins combined to the promoter regions of GMPS1s. And then we overexpress GM-ERF5 in the tobacco and use the promoter of GMPS1 to check if ERF5 can regulate this promoter. By luciferase analysis, we can find that GM-ERF5 can regulate the promoter and activate the promoter function of GMPS1. So we use the soybean hair root overexpress the GM-ERF5 and the GMPS1 to check if NOP-AA have response and can regulate these genes. From the result, we can see that when these 2 genes are expressed, the nodule number is increased, and other types of effector may be also involved in this signal pathways. And as is the protein-protein interaction, we also want to know if the want is a physical effect of this type of effector. So we test the hormone content and of this effector-induced genes. And by the iron cycle analysis, we found that some hormone and immunity-related genes and transcription factor also induced by the NOP-AA. And this transcription factor can regulate a lot of different kinds of genes, not only one, but may be involved in the hormone or immunity, something like this. So by the hormone content analysis, we found that NOP-AA also can regulate the ABA and something and the GA and related with the hormones during the rhizobium infection soybeans. And so in the same times, by the phenotype analysis in the soy chromosome segment substitute lines, we also found 2 different special lines. And one is F1680, another is F1011. These 2 lines we can see that have a reverse phenotype by inoculate with NOP-AA mutant. So it's meaning that some chromosome segment was substituted to response to NOP-AA to affect the nodule with reduced induction of NOP-AA. So you use this two lines and the wild type 314. We do the RNA-seq again to check what genes is different induced by NOP-AA, and also combine the QTR location result. We can select 3 chromosome regions on the chromosome 11, chromosome 7, chromosome 12 to other candidate regions maybe have genes response to NOP-AA, and we also clone the other to check other rhizobial mark genes to see if NOP-AA can affect the expression of these genes in these 3 lines. Finally, we identify that GMDRR is one candidate genes can respond to NOP-AA. By the RNA and OR expression length analysis, we found that GMDRR also is genes response to NOP-AA, and the NOP-AA induced symbiosis establishment also required GMDRR. So here we combine all the results. We give a result for NOP-AA is like that. When the NOP-AA in fact secreted into the soybean cells, first it can be directly interact with GM9 and GM-ABHD, and these 2 genes may be involved in other immunity signal pathways. And GMDRR is also not indirectly proteins response to NOP-AA to trigger maybe lignin in synthesis. And in the same times, NOP-AA can binding to the transcription factors EIR5 and to promote the ABA hormone side in synthesis to regulate the symbiosis. But we know that there's too many proteins. We need details experiment in next step to elucidate the mechanism of NOP-AA in regulating the symbiosis. So combine all the type 3 effector result, we add more signal pathways on the current signal network like this. For that, NOP-M may be ubiquitous NOP-M and GM-MIP in the soybean cell to involved in the ETI, and NOP-P interact with GM-PBS1 and the RN4 module to regulate the ETI, and NOP-C can interact with GM9 and the GM-ABHD involved in the ETI, and in the same time, NOP-A can also interact with GM-ERF5 and GM-PS1 in nuclear to regulate some genes expression to trigger the hormone synthesis, something like this. And for the indirectly interaction, we found that maybe NOP-P also involved in this network, and NOP-AA also involved in the NOP-C involved network. So this gave our clue that when the rhizobium infects the soybeans, the not type 3 effector is not a single one to play the function for the symbiosis establishment. There must be a multiple function for different type 3 effectors. Maybe they have a collaboration to trigger different soybean harbor genes to regulate the immunity or the signal pathways to regulate the symbiosis establishment. So recently, because we also construct a different single, double, or triple mutant or type 3 effector. By the construction of these type 3 effectors, we do the nodulation analysis on 314. We can see that when the TTSI is also a regulator to the expression of all the type 3 effectors in rhizobium. We can see that when this protein is a mutant, there is nearly no a significant shutdown of nodule number is found but one single type 3 effector mutant, they also have a different phenotype, maybe positive or negative. But when the double mutant or triple mutant was construct a very novel interacting phenotype can be found. This is nodulation result suppose that maybe like that nop L and nop P maybe have interaction, and nop L and nop C also have interaction. But this needed to fusion work to elucidate which is connected proteins for the two effectors. And in the same times, by the double mutant constructed with nop L and nop P, we also do the nodule phenotype analysis. We found that in the NGR234, nop L mutant can have effect on the nodule synthesis. But when this also happened in soybean, but when we know we construct the double mutant of nop P and nop L, we can see that this synthesis is inhibited. This means that when nop L mutant, then maybe the nodule can form can activate the synthesis signal pathways, but when nop P also mutant, this signal pathway is inhibited. This also need a future work to elucidate this. And in addition, as I showed before, the nop T can be interacted with GM-PPS1 and GMRN4B. But when we do the double mutant with the two effector, also reverse phenotype was identified. We can see that in another germplasm is Dono 15, you can see that this module number also very significantly different by the double mutant. So this means that this also have network interaction between the two proteins. And this similar function also found between another three type 3 effector, such as number AA, number T, and number C. So it's only for the triple mutant. In future, maybe we need to mutate all the type 3 effectors in the rhizobium to see what is really happened during the different effector triggered signal pathway. So here we give a model is like this. In recently, we only identify this 5 type 3 effectors. Maybe this type 3 effectors are involved in the immunity, but if the crosstalk between the different effectors, symbiosis can be constructed during these effectors interactions. So it's finally all the work is supported by the foundation from Northeast Agriculture and the China Scholar Council, and also the National Natural Science Foundation, and also the national key programs, and also science profession and the dog lab give me a chance to have learning and studying experience in Hong Kong, and also thanks all the students involved in my teams. All the work is done by them. So thanks everybody. My presentation is complete.
So this talk is a bit academic, but it's very important because when we say soybean is environmental friendly, the mechanism is related to the interaction of rhizobium. So maybe may I ask the audience to raise the question now? Okay, maybe I start. Okay? So yeah, so HH103 is Rhizobium Freddy, right?
Yeah.
Okay, so is this the one have restriction among different host or soybean host, or they don't? Because we also work on host range, right, of the Rhizobium Freddy before. And I just wonder whether the HH103 is like they can infect all hosts, or they are restricted by some of them.
It's a good question. Yeah. By the nodulation analysis in soybeans, we found that HH103 can success from nodules in a lot of soybean germplasm. But when we also found that some landrace or wild soybeans cannot construct a good symbiosis relationship with one HH103, maybe depending on the genetic background, I think maybe because by the nodulation for formation, we found that several landrace of wild soybeans only can form single or 12, 2 or 3 nodule, something like this, not so good. So, but most cultivars is normal for this strain.
Okay.
Yeah.
Because previously we're working on the restriction of some soybean to some rhizobium. So
Yeah.
I noticed that you have mentioned about the loop P. so
Yeah.
loop P in rhizobium, I believe is interacting with our RFG1, and loop P in rhizobium.
RG1. Yeah.
RJ2 is the interaction. And so I'm not sure whether the HH103 has which type of loop P, because I see when you have the mutant, you're actually increasing the nodule number, right? In loop P mutant. So, yeah. That's very, very interesting. Yeah.
Yeah, for the NOP P, because you recently, as I worked in your lab, I also do the NOP P use in the soybean. As RG2 is response genes to NOP P, but not directly interaction. In recently, in the Xu Wang's lab in China, he also used USDA 110 NOP P to identify the directly interact protein is the GMNNL1, also can directly interact with NOP P, but his result didn't find RG2 can directly interact with the USDA 110 NOP P, but in HH103, we also compiled the proteins sequence with alignment, and we found that the diversity between the HH103 or USDA 110 is different. So we think maybe in the single rhizobiums, the NOP P's function maybe have difference with the rhizobium. As we also do the IPMS analysis, we also didn't find the GMNL1 in our result. So we don't know why. Because in rhizobium, the interaction between NOP P and GMNL1 is performed, but in rhizobium, single rhizobium, we didn't find it. So I think it need more detailed work. Yeah.
Yes. Okay. So yeah. So is there any other questions from the audience? Please unmute yourself and talk. Quite quiet, huh? Yeah.
Yeah. Maybe too late.
No, it's okay. I think the talk series is more layman the past, and so there we have some non-specialists in the audience. But maybe may I invite you to explain, right? So how your research could help the cultivation of soybean or help the function of soybean? What is the path to impact? So after you do all these complicated level analysis, how could you make use of it?
Okay. As we do this research in Heilongjiang province, we also wish to apply the result in the soybean breeding. Recently, we analyze the function of this type 3 factors. We found that this type 3 factor is very important to control the nodule numbers for different cultivars. And so we think in future we can isolate here we didn't show the recent result is that we also isolate a different rhizobium from the field and use different soybean cultivar to arrest this rhizobium from different kind of soybean field. And then we inoculate back again to the field and in the greenhouse to see which rhizobium is the excellent rhizobium can have good nodule formation in the field. So after we sequence all the rhizobium isolated from the field, and we also wish to maybe do some small modification for different tapestry factors, then maybe can control the nodule number. As we know that the symbiosis is a nitrogen, is a nutrient, it is a repair nutrient from soybean to feed back. So maybe control the nodule number can control the efficiency of nodule fixed nitrogen energy experience for the soybean, and it may be good for the yield. So it's my idea for the future soybean breeding.
Okay, so I guess let me see if there's any questions by the audience. Yes, Joanna, please.
Very interesting talk, but I have to admit that there are many terms that I really difficult to catch up. But my question is, out of my curiosity, you see these they are needle shaped, that you have several pictures to show them.
Yeah.
You said that pathogens also have that. What kind of image is that? Is a microscope live or by some kind of fixation to get that kind of message? Image.
For the needle?
Yeah. Yeah, you have several photos to show the needle and injecting into the
yeah. All the needle is not from our lab. It's from the published papers. Because the tapestry effector is conserved in some pathogens. Can you hear me?
Yes, continue, please.
Okay. It's due to that, because we know that all the bacteria belong to the prokaryotic cells. They didn't have the gene function can be same as the eukaryotic cells. So maybe during the evolutions, the bacteria construct these systems only use several small amount of proteins can be hijacked the host system's proteins. Then during the infection, these proteins can be used as a soldier. It's secreted into the host to interact directly with some helper genes. So as you said, for the needles, the publisher also say that for the different bacteria, some needle, the tapestry secretion systems that belong to as like a needle. If the needle is long enough, it's not function as the needle. It's maybe like as a movement for the bacteria, not for the ejected protein. So it's the evolution for the bacteria, I think. So because this direction is not my research focus, so I only know this is the structure for the protein can be separated from the bacteria into the host, and maybe in future I can see what happened for the rhizobium.
Yeah, I think if you want to see the type 3 the structure, probably you need electronic microscope to see it, right? So type 3 includes cell interaction. So it's supposed to touch each other, and then the bacteria will pass the protein into the plant cells. So this is the interaction between two cells through a kind of a channel. So it's very difficult to observe without like a electron microscope. Even a light microscope, it could be quite difficult. But sometimes when we look at the whole bacteria, you see some pili, right? So but those are for movement. Yeah, but for the injection of protein, I think we really need a high power microscope. I'm not sure whether I'm pretty sure that people have done it, but I'm not sure whether some of your picture actually showing this type 3 system. So type 3 effector is go for a type 3 system. Type 3 system is a cell-cell interaction. So that I think that will probably explain the question about from Joanna, because I think she's talking about the structures that enable such an interaction. Yeah.
Okay.
Okay, thank you, Dr. Lam.
Yeah.
Thank you,
Okay.
Joanna.
Yeah. Any more questions? Now, so if not, let me do an advertisement. Okay, so I would like to advertise the next talk. So in the 31st talk, the title will be the fate of heavy metal in the soil environment and what is next. So the speaker is Dr. Li Wei Chen. Okay, so you're welcome to join us on May 23rd next month. Okay, so with that, I thank everybody for joining. So good night, everyone, and maybe good
Good night,
morning.
Dr. Lam. Bye-bye.
Bye bye. Bye bye.